and the human person.
Good afternoon, everyone. Thank you so much for joining us for this incredibly important conversation on editing destiny, the debate over human genetics. We live in a moment of astonishing scientific progress. Just a generation ago, mapping the human genome was a distant dream. And yet today, researchers not only read our genetic code, but in some cases edit it to heal disease. What once took many, many years and millions of dollars can be increasingly streamlined to treat deadly diseases. And that gives us a breathtaking opportunity. New gene and cell therapies are already transforming lives. Through the National Institute of Health's somatic cell and genome editing program, scientists have learned how to target diseases at their root, precisely repairing damaged DNA in living patients. Earlier this year, a child known as baby KJ became the first infant to receive a personalized gene editing treatment for a fatal condition. Instead of facing a lifetime of invasive procedures or an untimely death, he was cured. As one NIH scientist called it, we live in a new era of precision medicine. And this is precisely what real innovation looks like. As Secretary Robert F. Kennedy Jr. recently said, quote, "We are absolutely committed to making sure that the U.S. remains the center for cell and gene therapy research around the globe. We understand this kind of research is absolutely consistent with the MAHA agenda. These are technologies that cure disease. We need to move away from the sick care model where we are treating diseases over the lifetime of the patient and actually cure the disease," end quote. That statement captures the heart of what we call the Make America Healthy Again vision, using science to restore, not merely select, the human person. It is an ethic that sees innovation and moral responsibility as partners and not rivals. But not all uses of gene technology share that purpose. While researchers in hospitals and labs work to cure children like baby KJ, a very different movement has emerged from Silicon Valley. It promises parents that they can optimize their children before they are even born. Companies like Orchid, Nucleus Genomics, Harrisite, and others now market full genome embryo testing as a way to select the healthiest, smartest, or most gifted child. Noor Siddiqui, the founder of Orchid, told the New York Times in a conversation with Ross Douthat that her company gives parents the power to protect their children before pregnancy begins. By sequencing 99% of an embryo's genome, her team claims that they screen for thousands of disorders, and such technology can even be used for traits like intelligence or personality. My concern, and the concern that we will be unpacking today, is that such technology and such screening services discourage genuine medical innovation. When the goal shifts from curing disease in existing patients to preventing undesirable patients from being born at all, the incentive to develop treatments declines. And yet, even if embryo screening worked perfectly, it would still be wrong. It reduces human life to a list of potential traits. And this is the heart of consumer eugenics, the belief that we can design better people by rejecting the human embryos who don't measure up. And while it sounds compassionate, it covers a far more deadly and dark reality, one that we're going to unpack today. So this is where policy, ethics, and cultural conversations intersect. As a country, we're facing a choice. Will we invest in therapies that cure disease in the living, or in technologies that eliminate those who might one day be sick? This debate is playing out in labs, venture capital portfolios, and policy meetings, and it involves four main coalitions. First is the Make America Healthy Again movement, which sees gene and cell therapies as tools to treat root causes of disease and to make healthcare restorative rather than simply reactive. Second is the pro-family movement, which calls us to protect human dignity from conception to natural death and to uphold family formation as the proper context for family flourishing. And third is the Silicon Valley transhumanist movement, who views technology as the key to overcoming not just disease, but human limits themselves. They often speak of defeating aging or optimizing humanity. It is a worldview that seeks to control nature rather than live in harmony with it. And finally, there are the biotech innovators who are walking this very fine balance between both worlds, embracing scientific progress while calling for guardrails to ensure that healing remains at the center of human genetics. And so, the promise before us is incredible. Therapies that can cure, not just manage disease, innovations that align with human dignity, and a pro-family vision that resists the transhumanist dream of remaking humanity in our own image. Our task before us this afternoon is to ask whether we will use our power to edit destiny or to redeem it. Thank you, and welcome to Editing Destiny. It's my delight to welcome Rachel Roth-Aldheiser for a fireside chat conversation. So Rachel writes on culture, politics, and family life. Her reporting and opinion has appeared in the New York Times, Wall Street Journal, the Washington Post, and many, many places. Rachel's commentary was featured on both radio and television, and she is a visiting fellow at the Ethics and Public Policy Center and a recent recipient of the Robert Novak Journalism Fellowship. Welcome, Rachel.
Thanks for having me.
Of course, Rachel, thank you for being here. So I wanted to begin today's event not simply diving into the technical questions of regenerative gene and cell therapies and polygenic testing, but to really understand what's at stake here and the real live humans who were behind this. And I have long admired you and your work in this place, but not only do you approach this from the perspective of a policy thinker and ethicist, but a mother and an advocate and someone who has walked alongside your own son's life. And so I was wondering if we could start this conversation with you just taking us back to 2021 when you received the first ultrasound appointment that made you realize that this is going to be a very different experience than you expected. And particularly, I would love to hear about the early conversations with your doctors and how their words helped shape your approach to raising a son who does have genetic disabilities.
Thanks. Thank you. It is really a privilege to be here. Anytime I get to share about my son, David, I am reminded that I am a really grateful mom to be able to witness sort of the miracle of his life, and that it's a very powerful 4-year-old little boy who does not walk, talk, eat, or do much independently in the way we think of other kids, but he's powerful enough to have me come on this stage and be able to share with you about his life. So it's really my privilege to be able to do this for my son. So when I was 17 weeks pregnant with what at the time was our third child, we now have 5 kids, we went in for what we thought was a routine ultrasound appointment. And if you've ever gotten bad news at the doctor's office, you know exactly how that feels. There is a before time and there is an after time. And it feels like nothing about your life is ever the same. And so we learned in that appointment that David would be born with a set of very rare orofacial defects. At the time, our physician's best guess is that David would be stillborn or die shortly after birth. I'm here today to tell you that David is miraculously living. He is 4.5 years old. We are so grateful as a family to be able to witness his life and his story. And we are along on David's ride. So those early conversations, I was thinking about this on our drive up this morning, happened in 2 sort of very different veins. So one, my OBGYN and my maternal fetal medicine specialists had the same metaphysical priors that I did. Never did they once pressure me towards terminating my son or even questioning the value of his life. But my OB, who to this day is a very dear friend, the first thing he said to me and my husband was just, "You are the right parents for him, and you can do this, and I will be here with you." And as I said, if you've ever gotten bad news, what you need from your physician is not their prediction, right, for how the rest of your life will go, but you need their presence. And so I was blessed to be able to have physicians who were present with me on one hand. On the other hand, I had some very, very challenging conversations with specialists. And one thing we know is that when pediatric patients receive bad news, that news is delivered differently if the patient in question is a pediatric patient or an adult patient. Physicians tend to give the rosiest, best picture when the patient in front of them is an adult. But when that patient is a child, whether for reasons of liability perhaps, or just wanting to really prepare parents, physicians give the worst case scenario. So I'll never forget this. I'm not a physician, just a mom. I was, at the time, 28 years old, and I was on the phone with my son's, who would be his plastic surgeon. And I was like maybe in my 3rd trimester of pregnancy, and he was telling me, your son will be stillborn, and there's no way that he will survive. I'm looking at the MRI, looking at his brain. I just don't see this going the way that you think it will. And I remember walking him through step by step those MRI results, me without a medical degree, but having listened to a different perspective. And at the end of that conversation, he was like, well, I feel better about this. I feel better about your son's odds. So that is the kind of environment that parents are put into when they receive life-altering diagnoses. They are not doctors for the most part, and they are people who are very scared. And they're suddenly thrust into an incredibly intense emotional environment where physicians, there's a power dynamic there between the patient and physician. And I think what we see a lot is that physicians sort of take liberties with describing not only what they're seeing in terms of results, but in terms of predictions for how that will be for the rest of their life. So on one hand, I was very blessed to have doctors who were willing to be humble and honest and say they didn't know how my son's life would unfold. On the other hand, I've had a lot of very frustrating conversations with physicians who really wanted me to get on the same page as them.
Yeah, so today, some in the genetics world talk about using technology to really maximize choice and opportunity for children, as if love means selecting the best possible version of your child. So your story with your son, David, seems to tell a very different story. So how has being a mother changed the way that you think about unconditional love and choice and what it means to welcome a child as they are, even in this rapidly developing technological environment?
I think on one hand, we're living in sort of what I consider to be the golden age of disability support. My son is privileged to attend public school. He has a nurse assigned to him. He benefits from Medicaid, which I am so grateful. And I wrote about that for the New York Times several months ago. And the response to that piece has been really illuminating. The feedback that I got from extreme sort of progressive left-leaning folks was that I actually was making a very non-compassionate choice to bring a child like David into the world. I have letters to my house to prove it. I'm still getting emails every day for folks who are telling me that that government money would be better spent elsewhere, that because I knew that my son had a condition prenatally, that the responsible thing to do, the best thing for my son to do, would have been to terminate his life in utero. So I think as a culture, we are really struggling how to reconcile the presence of someone like my son, David. On one hand, we have these amazing benefits like Medicaid that provide really essential things for my son's life. On the other hand, we don't know what to do with the presence of someone like this because really it's a mirror. What do we believe about ourselves, our own value, and our own worth? Is it tied to what we can do, or is it tied to who we are?
Yeah, and so that gets at something I remember we were talking about with a doctor that you were working with after David's birth, and you asked a question along the lines of, how should I think about my son given this rare genetic condition? And I would love for you to unpack her answer.
Yeah, so, and just to clarify, so David has a constellation of really unusual birth defects. We are still searching for kind of what would be a genetic cause there. We don't have a clear answer, but we were working talking with a geneticist, and I remember just saying, what is the best way to refer to this in conversation? Should I call this a pattern of birth defects, fetal anomalies? Give me some guidance here. And I remember this is a highly trained medical geneticist, and she said, you can call this whatever you want to call this, that it really depends on sort of your value system as a person, how you want to approach this issue. And I knew right then that there was something really strange and unnerving about that statement, because what she was trying to do was make my son's experience as someone born with profound congenital abnormalities a subjective one, that it could be whatever I made it. That is not true. And I just remember sitting there thinking, this is the person in charge of figuring out more information about my son and giving us sort of a roadmap. And she's telling me that it's subjective.
Yeah, and I think what that really, reveals is that ultimately it's up to you as a parent, as an adult, as a born person to decide the value and purpose of the child before you. And I think that's where you very quickly move into this very dangerous realm of eugenics, where all of a sudden, if you as the parent deem that life not worthy or not important or not good enough, that you somehow have say over that. And I think that's where a lot of the tension seems to be playing out. So as we sort of wrap up this conversation, I was hoping that you could then paint a positive vision going forward. What kind of culture do you hope to see policymakers, everyday Americans build around genetics, not eugenics, but genetics and medicine? And how can we encourage innovation that restores and heals while also protecting the unconditional love and the sacred relationship between parents and children as they're navigating these conversations?
Yeah, so I think information can be a terrible burden. So when we talk to patients, are we considering if the information is actionable, and what kind of actions do we want to see those patients take? And are we giving them clues to what we think would be best for them when we give that information? Or is it sort of non-biased? How is it being used? And really today, this information prenatally is being used to make a choice of whether or not a child like my son is sort of worth the opportunity cost of bringing someone like him into the world. And so on one hand, I want to value amazing strides that we're making with things like gene therapies. We have a friend whose son has Angelman syndrome, and he is in a new trial that could someday really offer a cure or somewhat of a cure to kids like him. So on one hand, we're making these amazing strides, but on the other hand, why are we less able to answer some of the most fundamental questions of human existence? Like, does a person with a disability have a right to be here? Do they matter? So information and one can be a good thing, but where is it taking us? And is that a good place? Is that a place that I want to be? What will that place look like in 50 years? What kind of conversations will my children and their friends be having when they're my age, when they're parents?
Wonderful. Rachel, thank you so much for joining us.
Thanks for having me.
Our other panelists are welcome to join me on stage. And I'll begin the introductions as they're making their way up here. So we have, yes, you can sit in order there. Yes. So we have a game of musical chairs here. It'll be great. So we have Dr. David Prentice, the president and founding board member of the Science Alliance for Life and Technology. We have Timothy Hunt, the CEO for the Alliance of Regenerative Medicine. Dr. Todd Lintz, the associate director of the Feinstein Institutes for Medical Research and the professor of the Zucker School of Medicine. And then last but certainly not least, Dr. Benjamin Hurlbut, who is the associate professor of the School of Life and Sciences at Arizona State University. Welcome, gentlemen, to the stage. For this part of the conversation, we are diving more into the technical conversation around these developing technologies. So I want to start off with a question for you, Dr. Prentice. And I would love if you could just frame the broader conversation for us today. I know we're throwing a lot of new terms and new technologies at you, so we're going to just slow it down. And at first, if you could just help us see this conversation in the larger context of developing bioethics and biotechnology. It's been just over 2 decades since we first mapped the human genome. So how did that milestone open the door for today's regenerative gene and cell therapies, and also the rise of polygenic testing?
Well, it really set the groundwork. I mean, the goal of the Human Genome Project was to map the entire human genome, not just the sequence of the letters, of the bases, but also to determine what genes are present in a human being. There had been a lot of speculation, in fact, before the genome was mapped about how many genes a human being had. Did they have 60,000? Did they have 10,000? And so on. They finally got a number at the end, and it's a little bit north of 20,000. But the idea was to set the stage for potential treatments, therapies, and possibly how to intervene in terms of how our genome works together. Now, in one sense, that was a very simplistic way to look at things, that, oh, there's a gene for this and a gene for that. And we need to understand that that's not just simplistic, because many traits are multi-genic traits. They're affected by lots of different genes and genes interacting with each other. But it also sets the stage for how can you really know what to intervene and so on. There might be monogenic or single gene traits, but how do you know how these other genes work together and interact with each other? And I also want to take a step back in terms of the testing and so on that we've been talking about and will continue to talk about in this panel. Let's look at who is the object of that testing. We're talking especially on this panel about embryo testing, the idea of trying to predict the future of a particular individual. That's also a simplistic way to look at it, as if it's genetic determinism, that your entire future is laid out just within the sequence of bases. We already know that birth defects, what we've tended to call them at least in the past, only about half of them are genetically determined, that there are environmental cues and insults, if you will, during our development that can show up later on at birth. The other thing, too, is what is the real goal? In your talk with Rachel, you'd already alluded to this idea about choice. Choice of whether a particular human being should continue to survive. Choice of what would be a better human being. In fact, James Watson, of Watson and Crick fame, DNA helix, said back in the 90s, if we could learn how to manipulate genes, we could make better people. And one thing you ought to immediately be struck by is who's to say what's better? Every individual is a unique human being, a member of our species, no matter how old or how large they might be. And we need to consider that fact that that individual, if they're diagnosed whether as an embryo or after birth with a particular disease, that individual is not that disease or that syndrome.
Yeah, that's fantastic. So digging a little bit deeper into that, Dr. Lentz, I would love for you to talk us through an embryo report, as companies often call them. So based on the research that's been done so far, what can polygenic risk scores actually tell us? And what do people often think that they tell us, but maybe they really don't? And if you can define things like polygenic risk scores and embryo reports to go along with that.
So let me build off of what Dr. Prentice was just saying. Most of the diseases that we're interested in terms of diseases that affect most people at some point in their lives, whether it's cancer, diabetes, psychiatric illness like schizophrenia, these are polygenic. And that means not just that there are a number of genes involved, but typically that means hundreds, if not thousands of genes are involved. And what we've started to be able to understand better in genetics is how those genes combine to influence risk for disease. But these effects are very subtle and statistical in nature, so it's not deterministic in the sense of you have a gene for this disease, as you were just saying. But what we can say with a polygenic risk score is that out of all of the genes that contribute a tiny bit to the risk of a given disease, you have more of them than average or less of them than average. We could give that a percentile score. You have 95%, the 95th percentile, meaning most people have lower scores and you have a higher score of risk for diabetes, but maybe your risk for schizophrenia is at the 25th percentile, so it's lower than average. But again, that's all probabilistic in nature. The general risk to develop type 2 diabetes might be about 15 to 20%. So if you have a high risk score, maybe you have a 25 or 30% chance. But that doesn't mean you're going to develop diabetes, it just means you have a slightly higher chance. And you can affect that chance by diet and exercise and all of those things. So it's very hard sometimes to grasp the probabilistic and subtle statistical nature of these polygenic risk scores. But that's what these polygenic risk scores do. They assign a number to a thing that is quantifiable. Again, these traits have to have been measured in large-scale genetic studies. So what are the things that we can measure? We can measure diseases. We can measure your blood pressure. We can measure, even to some extent, IQ. But there are lots of things that we aren't able to measure and capture in a polygenic risk score, which often doesn't get discussed.
Absolutely. So a follow-up on that I would love to dig into. I think the big question is, you see a number of companies say 99% accuracy, you can trust us. And so the question I have for you is, how accurate are these reports?
Let me give an example. One thing that you can measure in a polygenic score is your likelihood to be relatively taller or shorter. We call it a polygenic score for height. The genetics of height are actually fairly well understood compared to a lot of things. And height is not fully genetically determined, but again, there are many genes that contribute to whether one is taller or shorter. We did a study in 30 very large families. These are families with 10 or more children. These children are now grown adults, and we know their height. So we said, well, imagine that these 10 adults were instead embryos, and I wanted to get a polygenic score for height and select which would be the tallest, because I want to have a very tall child. We found in these families where, again, the height was known, these are adults now, the individual in that family who has the highest polygenic score for height is only the tallest sibling about 1 in 5 times. About 1 in 5 times they're actually shorter on average than their other siblings and then their parents. And on average, much of the time, they're a little bit taller on average than their other siblings by maybe an inch or 2. So that gives you a sense in very concrete practice how accurate these scores can be. They're changing probabilities slightly, but that is very difficult to communicate in the clinical setting, and particularly in an advertisement.
Indeed, it is. So switching to you, Ben, you've written quite a bit that our debates about biotechnology often start with what's possible or what we maybe hope to be possible with the science, rather than the deeper questions of what kind of human future we want to serve. So how do we begin to reset that conversation, and what would it mean to build an ethical framework that doesn't just start with technology, but with human purpose?
So I get a softball for my first one. Well, look, so let me say something general, and then let me bring it back to this polygenic scoring thing as an example. So we have a tendency to react to technological developments. New technologies raise ethical questions, et cetera. And our conversations tend to follow that sequence. So we are now concerned about the use of polygenic risk scoring and what that particular technology might mean for whatever ethical questions it raises. But when you structure a conversation that way, it tends to make the technology the given, and whatever concerns about social and moral relations that you say it raises sort of secondary to the technology. Those conversations tend to be reactive. And if you think about this not just in terms of the structure of conversation, but sort of when the conversation happens and how that feeds back into what sort of projects get undertaken and what sort of concerns get contended with and when, it tends to be the case that the projects get undertaken before the concerns get contended with, because the projects raise the concerns. And there's almost a norm, you might say, that it's sort of, the presumption is, innovation is good. And if you can do it, you should do it, unless there are good reasons why you shouldn't do it. But to know whether there are good reasons why you shouldn't do it, you need to know what you're dealing with. And so maybe you need the thing that you're dealing with, or you need to have worked the kinks out of the thing that you're dealing with in order to determine whether it's a thing that, ought to be deployed. But by the time you've worked the kinks out of the thing, you've got the thing, and it's pretty much deployed already, and horse is out of the barn, and genie's out of the bottle, and the conversation's too late, et cetera. So you have this sort of weird dynamic where you go from, it's too early to talk about this because this is science fiction, this isn't technically possible, et cetera, to being too late to talk about it because now that it's technically possible, it was sort of too late. So there's this sort of dynamic. And I mean, we tend to kind of think about technological change and talk about technological change in these terms. Technology is moving so fast, society can't keep up, et cetera. I mean, to me, that's a kind of capitulation. Because actually, maybe society has been way ahead all along, because it's been contending with fundamental questions about, the right relations to our children, about human dignity, about the purposes and limits of medicine for a very, very long time. And a given technology can be fitted into those much broader, even if not technologically specifically delimited conversations. So take this polygenic risk scoring case, for example. Okay, to me, this is a little bit of just to be totally blunt about it, a little bit of a sideshow technology for precisely these reasons. I mean, it can't do very much, except what it can do is invite you to relate to your future children in a particular way, where your first relational act with your future child is to select it, right? And to think about health of your future child as somehow having priority over the individual to whom that health will be attached. So you're not worried about the health of a person, you're worried about health prior to the person, such that the person has to match the vision of health or whatever it is that you want. And to me, the right question about that is not, is this a good technology? Does this technology really do what it says that it does? What are the risks of getting it wrong, et cetera, et cetera? The right question is, this is a very fundamental human relationship, the relationship between parent and child, and for that matter, between medicine and patient. And we are enacting it in a particular way in this technological context. We should think about the technology in terms of that instead of the other way around.
That's fantastic. So I want to bring you into the conversation, Tim, who has been doing incredible work in the positive test case. So something that I've been thinking about a lot lately is how not every attempt to have a healthy baby is necessarily good, as our panelists have laid out, and yet not all gene editing is bad. And if you've been in the conservative movement for long enough, you sort of have this note of like, we're against gene editing and designer babies, and it's great in sentiment, but it doesn't fully account for the incredible opportunities that we have in this space. So I want you to bring that in. So help us set the stage here. When we talk about regenerative gene and cell therapies, what exactly do you mean? How do these treatments work? What kinds of conditions do they address? And why are so many people calling this a real revolution in medicine?
Yeah, sure. So maybe the starting point is to think about really disaggregating two different worlds. I know we'll come back to this, Emma, a little bit later on, but there's somatic cell editing. So it's editing on people that are alive today. Let's simplify it to people that are already born. Right? And then there's the world of germline gene editing, which is heritable, which is about embryos and things like that, right? So if you disaggregate those, the germline editing stuff is a whole, we'll get into that later on, just set that all aside. The stuff we're talking about are like medicines that are regulated by the FDA, in Europe by the European Medicines Agency, in Britain by the MHRA, right? So these are approved, essentially biologic medicines. We have things like different flavors, gene therapy that's been around, conceptualized since the early 1970s, and that's oftentimes the case where someone's lacking a functional gene. And so through engineering, we're able to take things like sometimes a virus or what's called a lipid nanoparticle, a delivery device, and you put the gene in there and you administer that to the patient so that they can recreate this protein. One example. Gene editing is things like, how many people, we may have people that are very sciencey here. I'm a reformed lawyer by training. I outsource some of my science to people that know far better than me, but many of us don't know that while we're sitting here, just listening to this over the last 20 minutes, our DNA is constantly breaking and repairing itself, thousands of times. I first learned this like about 10 years ago and I almost fell out of my chair. I'm like, what do you mean? My DNA is breaking like right now. I'm like, if you go up in a plane, right, your DNA is breaking and repairing itself much, much faster. It's a natural process. So gene editing is a system that some people have heard of CRISPR gene editing. CRISPR is what's called a nuclease, a form of a protein that gets engineered to help break your DNA by design in a certain area to turn off, oftentimes, a gene to stop a negative health consequence, right? So you can engineer things like that, again, heavily regulated by the FDA as a medicine, right? And then you have things like cell therapies, which is a simple way of saying they oftentimes extract cells, T cells, buff them up, make them sort of super cells, put them back into the body, and it allows you to fight things like cancers, blood cancers. Think of leukemia, multiple myeloma, and things like that. So the applications of these for cell therapy immediately are more things like these blood cancers I just mentioned. For gene therapy and gene editing, it's conditions like sickle cell disease, right? About 100,000 patients in the US with sickle cell disease. The average patient probably dies around the time they're 40 to 50, 55, oftentimes by a massive stroke. They endure many, many pain crises, oftentimes a year. It's a really horrible disease, right? There's rare things like spinal muscular atrophy. Maybe some people have heard of that, SMA, right? Affects children, they die at a very early age. There's a product called Zolgensma that was approved in 2017. It's treated, I think, about 4,000 of those patients. Many of them are alive today, right? Like, so these therapies, when they work well, can be life-saving, can be transformative. In some cases, the patients feel like they're cured. That's a very loaded word. We don't use that. We don't throw that word around. And I would say, for kind of fair balance on the technology, right? Like this generation of products, which are still fairly new, both in the US and Europe and around the world, no one says they're perfect, right? We've seen, if you go back in time, there was a high-profile death in 1999 to a young 18-year-old boy named Jesse Gelsinger. This took place at the University of Pennsylvania. This year, there were additional deaths that have taken place in clinical trials, right? So the technology is both very positive, very powerful, and still emergent and being studied, which is why we run these clinical trials. And these deaths, of course, are a real tragedy, but it's part of the process because we are typically treating lethal disorders, right? We know the natural history. A lot of the average patient population for the gene therapies that are approved in the US, the natural history would indicate that the average patient would die before they reach the age of 40 for all the drugs that are approved, right? So parents tend to know my child is going to lose functionality from Duchenne muscular dystrophy and die very prematurely. Therefore, they oftentimes reach for this choice, but it's a hard choice. So again, no one's saying these products are all perfect. They are not. But I think when they work well, they can be transformative.
So could you share, so we have the story of baby KJ over the summer, who I think is a very positive example of where these technologies are going and what they can offer. Could you share one or two other examples of where we've seen gene and cell therapies treat and maybe even cure or get close to curing patients over the last few years?
Yeah, well, I used a few examples. Let me give another one on the CAR T space. So there's a woman, some people may have heard of, Emily Whitehead, right? She was a young girl who developed a rare blood cancer and was really like written off for dead. And this is many years ago. And her parents really were like, this is pretty much it. with her. And they got her into a clinical trial for Kymriah, one of these early CAR T cell therapies at the University of Pennsylvania, right? They were just doing this clinical trial. She was the first patient dosed, right? And back then you had tough side effects. They didn't know how to manage the side effects that well. She went through all the hard side effects. They weren't sure if she was going to live. Today she is alive, and she is attending what? The University of Pennsylvania.
It's a good school choice for her.
She's remarkable. One other example is a woman I've been very pleased to get to know, Victoria Gray. Some people may have heard of her. She was the first patient dosed with a gene editing medicine for sickle cell disease. She was out at our conference back in October. We had her speak. I've heard her speak a number of times. I still get emotional when I hear her speak. There wasn't a dry eye in the room. At one point she said, very, very religious woman. She's like, look, I've got 3 kids. I'm having these horrible pain crises. I'm in the hospital all the time with sickle cell. Black woman in Mississippi. She goes in, she's traveling. She goes into an emergency room. I need pain meds. They think she's a junkie. She's trying to get on opioids. And she's like, oh my God, right? Like she's endured this for years, right? And she said, one night, I'm very religious. I just prayed. I've got 3 children. Lord, just take me tonight. I just want to die tonight. She's like, I hit rock bottom. She says that, there's no dry eye in the place, right? She's like, I wake up the next day, I'm still alive. Within 2 weeks, her doctor says, look, there's a clinical trial that's going on. I think this is not going well, and we need to consider getting you into this. So she's the first patient to be dosed in what was a CRISPR Therapeutics and Vertex trial. She felt great. She tells this amazing story. She's like, I woke up one morning. She's out of the hospital. She's back home. She's like, I woke up, my room's all white, and I thought, I think I'm dead and I'm in heaven. And she's like, because I woke up for the first time in my life, first time in many, many years, with no pain. And then she goes on to tell the story about how her life's been turned around, and it's like she feels cured. So again, it's case by case. In some instances, you have really rough outcomes, right? In some instances, you have almost miraculous outcomes.
That's absolutely incredible. And we'll come back to germline gene editing in a minute, 'cause that's certainly something that I would love to discuss. But Dr. Lentz, I want to move back because it seems that we're in this really interesting moment where we are, on the one hand, investing heavily in Silicon Valley and in other places in technologies that promise to screen embryos to avoid conditions or select for certain conditions, and yet are also finding out that there are incredible new advances in technology to actually help us treat those conditions rather than being forced into this decision of whether we're going to keep or discard. But one of the things that you and I talked about that I thought was really interesting, and Rachel alluded to this, is that it's incredibly important to consider how results are delivered and framed, even just in the order in which you offer the different options that you're scoring for or testing for. And so I was wondering if you could share with us a little bit more how the ways that we communicate polygenic findings, how that shapes real decision making by families, clinics, and even policymakers as we're considering these questions.
Well, I think that one of the essential ingredients of any medical decision and any relationship between a doctor and a patient is informed consent. There's informed consent to participate in research, but there's also informed consent just to receive certain treatments. If you go to the hospital and require surgery, they have you sign forms saying, I understand that I could bleed out on the table, that I might die, and so forth. Informed consent. We tend to focus in our society on the word consent because everyone's in favor of consent. But I like to focus on the word informed. What does it mean for somebody to be informed? First, there needs to be information. We need to know what are the probabilities and what do these genes tell us. And that's still under investigation. There's not a final story to that. The second piece to it is then, as you say, how do you communicate that? Communicating probabilities and even thinking in probabilities is really not a natural activity for the human brain and can become increasingly complex the more data that you throw at people. And we've written about this in some of our work, that there's something called the paradox of choice that you're familiar with perhaps in marketing. When on a supermarket shelf, there's too many choices. Sometimes a patient, sometimes rather a shopper, will just walk away and won't end up selecting any. And in fact, in the very first medical report, the very first published paper on this technology of polygenic embryo screening, the couple received their report, and there were 5 embryos, and 2 of them were listed as being at high risk for breast cancer. And the other 3 were not listed as being at high risk for any of the diseases that were available to be tested at that time on that report. But they actually reported in the paper, and I was surprised that the company would report it this way, but they were honest about it. They said the couple actually decided not to implant any of the embryos because they were so sort of spooked and thrown off by the whole experience of receiving the report. So one thing that I often will say is that informed consent is not just what the doctor informs the patient, but it is what the patient is hearing and how they interpret that and understand that. And that's something that we work on a lot in our research and is still unresolved in this area of polygenic screening.
So how are medical societies talking about this technology, and what is the guidance that policymakers, that people who are interested in like the application of this, what should they be keeping in mind, especially given the fact that there are many like yourself who are still diving into the research around it, and maybe others who have come to more firm conclusions?
I would say that every medical society that has issued any guidelines or statements on this technology of polygenic embryo screening, the American Society of Reproductive Medicine, the American College of Medical Genetics, and many others. I'm part of something called the International Society of Psychiatric Genetics. These societies have issued statements saying that this technology requires further research and is not ready to be administered outside of a research context and outside of much further and more careful study. That said, there is no regulatory mechanism in the United States that governs this technology or any aspect of IVF or reproductive medicine or assisted reproductive technologies. I could get into, if you'd like, at this point or later, the regulatory history in this area, but in terms of genetic testing broadly, and in IVF in particular, in the United States, there is absolutely no governing authority from the FDA or elsewhere.
Yeah. So then, Dr. Prentice, even if polygenic testing was 100% accurate and successful, would it be good for society? How should we think about it? I have frequently used the phrase consumer eugenics to address the market aspect of this, but how do you think about this issue in defined terms for perfect clarity's sake?
My short answer is no. It's not a good thing because it does make us consider children as just products that are being manufactured. You know, and further to what Dr. Lentz was talking about, there was a paper published just a couple of months ago about this whole idea of choosing embryos and what the patients hear versus what the doctor is trying to give them in terms of information. And it talked about how when the patients hear this is the best embryo in terms of all of the testing, and let's say that embryo is transferred but doesn't result in a pregnancy and a birth, then the parents are left with, so we're stuck with the second best embryo. And the idea that, you know, our children are sort of ranked in terms of value based on their testing scores. So, you know, it really is a situation where, number one, there's precious little to no regulation in terms of this, even in terms of marketing and consumerism, but also our simple attitude about how we view another human being, especially one that's a nascent human being.
Ben, I would love to have you jump in here. I saw the brow furrow, so I'm sure there's lots of good insights. But yeah, I'd love to hear you answer that question and also think specifically about the market company side of this, which is an additional thing outside of even a research context.
Okay. So it probably doesn't need pointing out that reproductive medicine is a business, but reproductive medicine is a business. I, a few years ago, spoke at the ASRM, the American Society for Reproductive Medicine, in this convention center room that was, it's like 6 football fields or something like that. It was insanely large. And there were a few little people sprinkled around here and there or whatever. We were talking about heritable genome editing. But then later in the day, there was a keynote that was another keynote panel that was called IVF is a Business. And it was packed. This room was absolutely full. What's that?
You didn't draw as well?
Sorry?
You didn't draw as well?
I didn't draw as well. No, I wasn't nearly as interesting. I was with Feng Zheng.
Sure.
He didn't draw as well either. So I think, one has to appreciate that. And, it's like the polygenic screening in principle, at least, is the Coke that you sell with the burger or whatever. It's you make a good margin on the extra. There's a lot of routine genetic testing in IVF that has basically been demonstrated to not only be essentially irrelevant, but actually to reduce the success of getting a pregnancy, where its purpose is to enhance the success of getting a pregnancy. But you make a few $1,000 off of it, and so you don't want to think about it too hard or ask too many questions or whatever. And so I think appreciating that is the context in which you ask for the informed consent of the patient to participate in whatever technology you're offering them, where it's their first time and you've already done it 9 times that day. I think that appreciating that context is really important, because there is a market for the kind of technologies that we're talking about. If you think that as a responsible parent, you need to go through polygenic screening in order to ensure that your child, whichever child you choose, has the best shot, and you don't need to go through IVF otherwise, you go through IVF in order to access the polygenic screening. When we talk about heritable genome editing, it's the same thing. So in a sense, you get the people to buy the burger in order to buy the Coke. And that is a vast expansion of a potential market beyond people who are seeking fertility treatment because of infertility. So I think it's very important to appreciate that. One other thing I want to say in relation to this, coming back to the informed consent point, I think one of the really compelling things that Rachel said earlier that we should hear is the way that the kind of relational interactions in her experience, but her experience is not unique in terms of how her situation was being narrated to her, how she was asked to make sense of it as she would or whatever. This has back of it this notion of neutrality, that the medical establishment supplies the information in a sort of neutral and non-directive way, and then it's up to the individual to figure out what it means. But that's also a way of shifting responsibility. And so I think we have to ask a prior question about whether there are responsibilities that are getting produced by technologies that are made available to medical consumers that basically burden them with responsibility, where we should be asking the prior question, is that a good responsibility to have? Is something good, something therapeutic, something beneficial, something, some moral improvement taking place by virtue of creating a situation in which a question can be asked that couldn't have been asked before? Or is it simply a kind of a cop-out that makes room for new products and the profits that come with them, and sort of displaces the problem of whether they ought to be there in the first place onto the shoulders of the person who finds themselves in the position of consumer, who, there are people who want to optimize their children, but it seems to me the real market for this kind of technology is people who want to do right by them. And anyone who's been a parent knows you really don't know what the hell you're doing once you get started in that enterprise, and you're trying to struggle to figure it out and do the best you can. And if you're told, here's a suite of technologies that allow you to do better, well, you're vulnerable to that responsibility.
So if we were to give you a magic wand to create a new social compact, as you've said before, what is the ideal, the positive vision for how we approach these conversations that you would like to see? And keep it, at maybe a higher level for the purposes of this conversation, but what is the better way to approach this if we got to restart all of these things and begin just with you?
So I'm going to give a simple-minded answer to that, and that is that, to me, the kinds of problems that we're talking about are relational problems. They are social problems in the most fundamental sense. After all, genetics is not individualist. It is relational, right? It is about interconnection in a sort of fundamental sense. And so we have pushed, and regulation of reproductive technologies or non-regulation of reproductive technologies is sort of exemplary of this. We've tended to push these problems into the domain of the individual. And so it's like it's a collective problem that is dealt with over and over and over and over again by individuals who have no kind of prior capacity to think about it. And it seems to me that just pointing to the market as the sort of space in which that problem gets solved is just doubling down on that sort of individualist orientation. To me, they need to be named as collective problems. They need to be contended with as collective problems. And those orientations need to inform the agendas of innovation themselves. If a society can say, it would not be good to foster this kind of relationship with one's children through whatever new technology, doesn't matter what kind it is, well then it's discouraging that domain of innovation, as opposed to saying, let it all come and we'll contend with the problems once they arrive, which is, you're having to play defense, you're having to react to an already established order. How to achieve that? I mean, to me, that is a political challenge in the best sense of politics. It's a collective challenge. It's a challenge for the polis. How to achieve that? What are the right institutional forms? Well, that's a more complex conversation, but just sort of capitulating and saying, "Oh, forget it. It's impossible. It's not even worth trying." Then we've sort of lost the battle before we've even started.
But if I can challenge you on that, precisely in that, how would you have such a conversation given the current attitudes tend to be fairly both pro-technology and laissez-faire when it comes to reproductive decision-making? We have published a study, general population study, a survey of over 1,500 representative Americans, demonstrating that between 75% and 80% support the use of this technology for the purposes of reducing risk for disease. Other people have published very similar results, so the surveys seem to be fairly consistent, at least in the US, that the large majority of people are at least open to this idea and intrigued by this idea. And when I give talks on it, people will come up to me afterwards and say, well, my mother had breast cancer, my grandmother had breast cancer, my sister has breast cancer. I want to have children that aren't going to have breast cancer, or fill in the blank, schizophrenia, or whatever it might be. That's a common response that I get.
Look, I mean, I think we have a thoughtless habit of assuming that all innovation is good innovation, that we need to sort of create an unfettered space in which innovation can happen, et cetera. And yet, even in that unfettered space, people are making choices. This is a project worth focusing on. This is not a project. This is a good thing to do. This is not such a good thing to do, right? So choices are being made. What are the contributions? What's shaping? How, individuals starting companies or undertaking research in their labs, how are those choices being formed? I go to a lot of scientific meetings, talk to a lot of scientists who are actively asking those questions and also asking, where do I go to ask these questions? I don't know. My ethics oversight committee says, yeah, you're not breaking any rules, so you're okay, go for it. But that's not satisfying. So to me, I mean, I teach college students. If I were to take a snapshot of what people think at the beginning of class and say, well, this is the landscape and there's the polarization, or here are the, and just call it a day, I would get a fundamentally incomplete picture, because you take the same snapshot at the end of a 45-minute discussion and you have a different picture. And it seems to me that we're skipping that 45 minutes. I mean, we are capitulating. We haven't built the deliberative capacity to actually ask these questions in a robust way, such that we can get past our sort of thoughtless presumptions to fundamental questions that, in my experience, I suppose this is research that I ought to get cleared with my IRB, but I make a habit of talking to whoever I'm sitting next to on the airplane about these kinds of things. And it takes about 2 to 3 minutes to get past these kinds of initial truisms. And then you actually are having a conversation, and there is a remarkable amount of overlap in the diversity of at least my airline companions, in terms of the basic fundamental human concerns that they would want to bring to such a conversation. So the challenge is to get it going, not to arrive at some result from it. I think that will take care of itself.
Fantastic. So I want to jump into the heritable gene editing discussion. And Tim, you were quoted in an article in the last few years where you said that, quote, human heritable gene editing is clearly a terrible solution in search of a problem, end quote. So this was in the context of the Chinese researcher who created the first gene-edited babies in believe it was 2018, if I'm remembering correctly, which really shocked the world and our response to this. But I want you to talk to me about the million-dollar question that this prompted within you. So why do you believe that germline editing crosses a line? What is the million-dollar question that really spurred your thinking around the moratorium and other approaches? But what are the problems with germline gene editing? And then how have you thought about how we wrestle with this from the policy and cultural perspective moving forward?
Yep. Yeah, well, the quote sounds like me. I'll take it your word. I was in Hong Kong when that news got announced in November of 2018. And it was for those that were there, it was a pretty searing, really unforgettable moment, right? The sort of red line that we all thought had been articulated suddenly got crossed with a real live person saying, I've just done this. Yeah. And it was, for those that may not remember, it was a big international scandal, like for days and days across Asia. We were in Hong Kong, of course, across the US, was in all the media. So it was a big deal. And why is that? Like, it goes back a little bit to what I remember at the beginning. I said, let's disaggregate these two worlds, right? There's the one world where you're making a medicine for these somatic cell gene therapy and gene editing, and then there's this germline world that is, it was at the time and remains today prohibited in the United States by something called the Aderholt Amendment. It's prohibited across all of Europe by the Oviedo Convention, which was essentially born out of some of the eugenic horrors people saw post-World War II. It's prohibited across most of Asia and many places around the world. And why is that? Well, it gets at a lot of the things we're talking about, right? It picks at things like, in this case, it was somebody who said, hey, I'm going to try to have people that are one parent's HIV positive and they want to have children, and we want to try to change a gene, edit a gene in the earliest moment of conception to make them not able to develop HIV, right? Which was a huge theoretical idea, no real work that had been done, no IRB that had been cleared, so highly challenging, right? So when you step back, we did a conference in March with our group, the Alliance for Regenerative Medicine, the American Society of Gene and Cell Therapy, and the International Society of Cell and Gene Therapy. So we brought together prominent academics, patients, bioethicists, religious leaders. We had a panel on religious views on this kind of thing, biotechnology executives, and we talked about a lot of these topics. And really the question was, what can you only uniquely do in theory with heritable genome editing that you could not do with somatic cell editing, either today or likely in the years ahead, given the pace of travel of where the science is going. And we really were like, it was reduced to, you probably could fit one or two applications into a very, very, very small thimble, like theoretical stuff, even if you could do it, right? But then when you went back, we heard from scientists that said, I don't know how you could do this anytime soon, right? Like, you can't just go to a legitimate regulator like the FDA or the EMA or in some country and say, well, I want to do it. They said, well, hang on. For any drug, you've got to show lots of preclinical studies, so animal studies. You've got to show lots of other work that you've produced, right? What are the exact animal studies you'd have to put together? Well, you've got to test it on things like non-human primates, monkeys and things like, well, okay, what do you have to show? Oh, well, you have to show many generations that you could do this and it would safely pass to the next generation, because once you make this kind of edit that we're talking about, it passes to all subsequent generations, which is an important point. So you make a mistake, it doesn't die with the person. If they reproduce, it goes on and on and on. So that's one of the reasons why it's considered a big red line, right? So scientifically super complex, ethically, it's a lot of the questions that are being discussed here, like, well, wait a second, like where would you stop, and what are the governance areas, right? Like, what's the difference between trying to improve health versus trying to enhance people, intelligence, height, looks, things like that, right? So it definitely gets into the eugenic world really quickly, and there is an endless series of questions that nobody really can answer. So our groups got together, we had this conference, and then we reflected on everything that we heard, we thought about it, we reached back out to some of the experts, and within a month and a half, we came back and said, we think there ought to be a 10-year global moratorium, that nothing should go forward in this area for at least a decade. From everything we can tell, the science is not ready. Even if the science were ready, there is fraught with ethical questions that really need to be thought through. And there are no governance structures to basically call balls and strikes on what should be allowed and what shouldn't be allowed. So we think there's great caution that ought to be put forward. There's been some articles that have come out lately even the last couple of weeks about 3 companies that have been formed. And I don't know a ton about those companies, but what I will say is if you hear anybody running around promising great outcomes from human heritable germline editing, I would just say, be wary of people selling snake oil, because they probably got some snake oil in their bag they're going to sell you next.
Yeah, so this is great. Dr. Prentice and even anyone else on this panel, what are some of the companies that are launching into the US market, the international market right now? A lot of them are being funded by Silicon Valley venture capitalist firms. What are some of the companies? What are they promising to do?
Yeah, and I don't know that I can even remember all of them presently, but one that keeps coming up by name is Orchid, which wants to sequence or at least do genetic analysis of your embryo and then promise we will give you the best embryo. And we've already kind of covered this whole idea of valuing and changing values. The ones that are interested in actually then manipulating and enhancing your embryo, one of them is actually called preventive, because their selling point is you're going to prevent any particular diseases or traits that you might not like. Because we also need to realize this isn't focused just on disease issues and disease genes. They're looking at actual traits, and you kind of see this sliding scale of what is a disease versus what is a trait versus what is just a desired capability or character and so on. But again, as Tim just said, if somebody says, hey, I can promise you that 99% probability you can add 3 or 4 IQ points for your embryo if you pick the right one. Dr. Lentz has been very elegant in talking about how, well, maybe on average, statistically, if you made the right pick, but there's also a range, and you might end up with 20 points down in terms of the IQ, thinking that you were going to get something. And it kind of brings to mind, again, this whole idea of consumerism. Are you going to get a money-back guarantee that if I don't get my kid, into the Olympics, or, they win a Nobel Prize, that am I going to get my money back from all of this? But it's a very consumerist attitude, and I like what Dr. Lentz was saying in terms of the informed consent. Parents are not being told, especially by these screening and enhancing companies, about what's the real information, what are the real facts involved. And that again affects our attitude towards not just embryos, but any human being in terms of how we're going to view them and their worth.
I will just add that there are actually 4 companies that are now offering polygenic embryo screening today. So heritable gene editing is on the horizon, and there are companies that are actually out there trying to develop those technologies. But today, there are 4 companies that are offering polygenic screening. And one of them, you mentioned Orchid. The CEO of Orchid, Noor Siddiqui, who you also, Emma, talked about in your intro, I had a conference on this topic a few years ago, and she was suggesting that her view of the technology is like, she made the analogy to online dating. That when online dating first came out, and I remember this 20, 25 years ago. That was kind of a very strange thing, and people thought that's really weird. Like, who would ever date somebody online? That's like the opposite of what you would want to do. And she showed a graph of how, you know, in the 2000s and 2010s, almost nobody used it. But then at a certain point, it shot up and it became higher than meeting somebody at work or at church or in a bar or whatever. It's the predominant way. And I have no crystal ball. I have no idea whether it will take off or die out. But that is her analogy that she provides for this technology as to where she thinks it's going.
So you're supposed to swipe right for your future child. Can I say a couple words on this? So let me just point out a couple of things that I think are, one thing that should be obvious, but it's worth pointing out. We do not allow clinical trials of risky technologies when there is no chance that the patient will benefit from it, basically, right? I mean, you got to have some potential for benefit if you're going to expose people to risk. That seems pretty straightforward. Heritable genome editing entails the genetic intervention, the introduction of the editing machinery at the moment of fertilization, basically, okay? So you create, so to speak, the patient in order to receive the therapy. To me, this isn't medicine, right? And it's really hard to wrap one's head around the idea that it would be acceptable to bring into being human beings in order to serve as experimental subjects in an experiment of the sort that Tim was describing a few minutes ago, where just practically speaking, the experiment is sort of crazy impossible to achieve a sort of standard for measures of safety and efficacy. But just on the sort of most basic, mundane sort of ethical standards that are used, somehow we can just trip right over, we just sort of skip past this detail that there isn't a medical need. There is not a patient looking at you that needs a treatment. You're creating the patient because you want to try the treatment out. That's basically okay, so can I say a couple of other things? Okay, so that seems to me points to the fact that the veneer of medical discourse that sits on top of heritable genome editing needs to be pulled back, and we need to look inside and actually see what's in there, okay? So I was also at the 2018 summit, and as it happened, I had met the Chinese researcher, He Jiankui, who produced the Chinese CRISPR babies. And I ended up having a series of very extended conversations with him while he was under house arrest before he disappeared into a Chinese prison. He basically didn't talk to, he told me his story and nobody else. So one of the things that he said in the course of that was that the rationale for undertaking this, and he really actually got a lot of affirmation and encouragement from a pretty wide network of fairly prominent scientists in the US and Europe and so forth, because he wanted to sound them out to, is this a thing that's worth doing or whatever. He got encouragement in his sort of rationale, and the rationale he got from others is that you have to push the technology forward in order for people to kind of get used to it. In order to, you have to have the breakthrough first in order for the kind of ethical realignment. In fact, what he said was, if you're waiting for a social consensus, this is almost exactly a quote, if you're waiting for social consensus, it's never gonna happen. But if you just go ahead and make the first kid, then science, law, ethics, society will all speed up. So it's actually about a catalyst for social and moral realignment. And boy, does that strike me as a backwards order, right? But if you think about what these emergent companies are, they're in a sense that. They're maverick operations that are wanting to push a boundary that's not just a sort of technological or biological boundary, it's a moral boundary. And do we authorize them to do that just because, whatever, they're companies and it's the private sector and you can sort of do what you want in the private sector, or because they can take their experiments offshore, as at least some of them, like preventative, are talking about doing, and so forth? It seems to me that the answer to that should definitely be no. Because if bad things were acceptable things, if done by private actors using their own capital or whatever, and none of us should have anything to say about it until, whatever, they hit the market, it's a very incomplete vision of governance. Last thing. Last thing. I think that the Chinese case is extremely instructive, okay? The discussion for years, and this is now a more than decade-long discussion, was about, we have this tool, is there an acceptable application for it? This scenario, the cases in the thimble, right, that Tim talked about. That's been we have this tool, can we apply it? Can we find a justified case for applying it? Not what are the parameters within which we should think about what it means to have this tool, but rather, is there a doorway we can find that allows us to go through from the not doing it side to the doing it side? And then we'll figure out where we are and what's on the other side of the doorway once we're there, right? That was the sort of structure of the conversation. But as Tim was saying, there are not very many things that you would put in the sort of domain of medicine straightforwardly that fit into this. But it simply is a fact that you cannot use polygenic scoring or monogenic scoring or whatever to select embryos that don't have a trait that you want. The only way to get that trait in there is to put it in there, so to speak. And in the Chinese case, the variant that He Zhenkui was trying to create didn't exist pretty much in Chinese populations, but it exists in certain Caucasian populations. He said to me, it's not fair that Chinese don't have this trait that's beneficial in certain ways. I mean, so the benefit is that it confers, in a sense, it confers immunity to HIV infection. The couples that he enrolled in his trial the criterion was that the male is HIV positive, okay? HIV is controlled on antiretrovirals, but is HIV positive. Being HIV positive in China is a very, very stigmatizing social condition. Even if biomedically speaking, it's not all that consequential, right? And so these couples that participated in this trial were wanting to, in effect, genetically inoculate their children against the social stigma that would come from being the child of an HIV positive person, since it's the whole family is sort of marked, okay? So this was the first genetic intervention in the human germline was a genetic fix for a social situation. And you can understand why those parents would think that that was the right thing to do. Because what can they do about society's attitudes? But what they can do is avail themselves of a technology that is there before them. And maybe they should. And I think putting people in that position is unconscionable. And that is a social failure. It's not a failure of those individuals, it's a social failure. In fact, the couple wrote a letter to the court during the criminal prosecution of He Zhenkui, at some risk to themselves, I think. And the letter said, we knew that there was a way to avoid HIV transmission in a sort of medical sense. There's a cure for that, but you cannot cure prejudice. So this was a cure for prejudice. And it seems to me this is exemplary of the kind of thing that people will seek in the emerging genetic repertoire if we pass through that doorway. It's not the monogenic conditions, it's these kinds of things.
Okay, final question, 30 seconds each. What is the most important question no one is asking right now?
Let me go?
Yeah,
Yeah,
sorry,
so
Tim.
I think I'll give you credit for this, Emma. Going back to our first conversation, I think, not your words exactly, but how do you get the balance right between putting these needed guardrails on some of these frontier technologies, and there's a basket of them. We could talk about them all day, right? It's a sliding scale, I think, but way out there, I would argue, is germline editing, right? How do you put needed guardrails around these frontier technologies on the one hand, while on the other hand, unleashing the power and the potential of real cell therapy, gene editing, and related technologies to help people for whom time is too off in life, right? You're a parent, you got a kid on the clock, they might die within a year, baby KJ. They might die within 10 years, right? They might die because we didn't get into this today, but in China, they are extremely focused on developing these therapies fast and at very low cost. It's a big global race on the somatic cell side. And so I think we need to continue to think about innovation in that area.
Dr. Prentice?
More than 30 seconds.
And Tim stole my guardrail, I think so. But anyway, I think on a wider view, how do we educate society in terms of our viewpoints of any human being, whether they have a disease or not, whether we're trying to create them in a certain way according to our desires, but how do we get that conversation going and actually change hearts and minds?
And I would kind of take both of those points and also add, how do we situate that in a global perspective when somebody could travel from one country to another for a medical procedure that is or is not available in a given jurisdiction, and when attitudes in some place like East Asia or Singapore, for example, might be very different, or in Germany or UK relative to the United States?
To me, the most fundamental question is a pretty simple one, which is, should we want this? Is this good for us? And we are not good at asking that question as a collective, as I think you've learned this is my hobby horse. We need to get good at asking that, because the potential for good in this domain is absolutely vast, but only if it is nurtured and guided by a surrounding sense of what is good for human beings, what is appropriate to human integrity and dignity, what are the aspirations to heal, to improve life that are worthy, and what go beyond the bounds or are transgressive against what it is that we're trying to protect, because the condition of being human is not a disease in need of a cure, right? And so I think that asking that question and developing the capacity to ask that question situates this domain of innovation in a kind of on a moral foundation, which is absolutely critical. And then, if other people elsewhere in the world do something different, we don't need to compete with them. Like, we don't, there are human rights violations elsewhere. We don't say, well, we better get into the human rights violating business. And yet somehow that is the sensibility with technology, like it's a race to the bottom. So if we can embed our projects of innovation in a more robust, more kind of ongoing, deliberative, moral conversation, we'll be much better off.
So now we turn to audience and online questions in the few minutes that we have remaining. So if you have a question, raise your hand, and one of our lovely interns will bring you the microphone.
Do you want to call on someone?
Oh, sorry, yes. Here, we can start with Caitlin and then work our way back.
Quick answers.
Yes, and quick answers for the sake of time. And if your
Yeah.
questions could be a question, that would be fantastic.
Hi, my name is Caitlin Shelton. Thank you all so much. I'm curious what you would say to people who say, well, China is going to outcompete us, and we need to hurry up and not have guardrails because it's a matter of national security, and we need to beat them to this technology.
So quick, in the spirit of quick answers, I think China, in our world, we heard very little about China 2 years ago. Things have changed very dramatically. The quality of their science has increased. They've got many, many clinical trials that are taking place. The China threat is very real. The way we think we can best compete is to just continue to streamline how things are done at the FDA primarily. A little bit of how you could do things with CMS to maybe better fund cell and gene therapies, but keeping needed guardrails in place. You still want to have clinical trials. You still want to really move things along through an ethical framework to develop somatic cell medicines, but the timelines are still very long. And to his credit, Martin McCary has talked about this. I think they're trying to think through how can they execute that, and we and others, I think, should probably play a role in trying to help that, because that's the antidote in my view.
I'll add one thing to that, and that is in total agreement with Tim, and there are ways of arranging things in this country that we arranged way back when for other reasons that worked pretty well, and they're not working very well in this. It really warrants serious assessment, because there is serious competition in this domain. But part of the reason to maintain scientific and technological leadership is to maintain moral leadership. And ideally, scientific leadership should be moral leadership, because science should be an expression of the values of the society out of which it's emergent, right? And so I think that it's crucial that we not become fixated on technology racing ahead, to hell with everything else because we don't have time to think about it. But actually, that may be racing down instead of ahead. That may be racing backwards.
To the bottom.
Yeah, absolutely. Okay, this gentleman here.
Hi, Walter Weber, American Center for Law and Justice. My question is, what is the difference between genetics and epigenetics, and how does that bear on the predictive capacity of these tests?
But genetics refers to the actual genes, the DNA sequences and so on that we've been talking about, and whether they're screening for, adjusting and so on. Epigenetics is what is expressed from a gene. So probably 90 to 95% of the DNA within any individual cell is not expressed as a protein or as a functional RNA. That still, though, depends on what the overall makeup is of the genome. And you can change, though, the epigenetics with various ways of treatment, different chemicals, different stimuli, and so on. And it actually puts the burden back or puts the focus back on the truly therapeutic things that Tim's been talking about, whether those are cell or gene-based, that if you manipulate the epigenetics, you don't worry about, well, we've got to change this individual from conception on. You can focus on the actual problem or condition that you need to be correcting.
Okay, time for one final question.
Hi, thank you for your time in this talk. Just thinking about gene editing as a medical solution to a lot of medical issues nowadays, and with the topic of, I would say, like autism, in terms of like knowing that Fragile X syndrome causes a lot of symptoms of autism, whereas just a few weeks ago, or not a few weeks ago, but weeks ago, I guess the discovery of acetaminophen, or like Tylenol, also being as a possible cause of autism. Do you see, I guess my question would be, will future medical treatments prioritize gene editing approaches or improving pharmaceutical drugs? And what challenges might prevent gene editing solutions from being a more popular approach in just the medical field today?
Well, I think that there are definitely many autism, again, is another condition in which there are many different genetic features that can go on, as well as non-genetic features that can contribute to risk. I think, Tim, I don't think you would disagree that genetic therapies, gene therapies, direct germline or somatic cell, rather, editing is much more complex. It can be potentially curative, essentially, but it is very, very complex, fraught with a lot of hazard to the patients. So I don't foresee it as overtaking the market relative to regular biologic and molecular medicines, whether for autism or for any other condition. That said, there are no molecular or biological treatments for autism. So it would not be surprising to see people attempt to approach somatic cures. But if it was for individual patients who have that genetic indication, such as Fragile X. Most patients with autism do not have Fragile X, although Fragile X is a potential cause of autism.
Yeah, exactly. There's a small basket of these more genetically defined diseases that appear to have a role with autism, and there are some clinical trials that are going on. I think they've begun or they're underway. A lot of the challenge, though, and I'm not the scientist on the panel, but if you think about it, a lot of what is oftentimes a rate-limiting factor in getting after any of these diseases, what is causing the disease? We don't understand the biology upstream well enough to say that's how we can intervene. I've got a son with a rare disorder called Dandy-Walker syndrome, and among other things, he has intellectual disabilities, and his syndrome was discovered a little bit like Rachel's, and my wife was 18 weeks pregnant with him, and it relates to the formation of the brain. Well, what's the constellation of genes that clearly are understood to get at the formation of the brain? Will we know in 10 years, maybe 25 years better, right? Like autism, what are the exact genes and how can you intervene with them, right? But do you really understand upstream what's really driving the the underlying issue. That's the problem with lots of these disorders. And it gets into things like, when in the germline world, okay, well, I want to have, if somebody wants to have somebody with a higher IQ, same thing, right? Like, how would you even begin to try to unpack that problem? It's so complex.
Please join us in the foyer across the way for reception after this, and join me in thanking our wonderful panelists today.